Let me first put my title on. This talk sort of evolved in a while, and mostly prompted by Eugene Maya, who was part of the CMU activity, but that never happened. And then the same sort of theme here. What are we doing at NASA in the search for life that's relevant to information science? And so I'm really mostly asking for help in some sense, rather than giving you answers. And I'll explain why I think it's useful to think of the origin of life and the search for a second genesis of life on other worlds in the context of a problem in information science. And to basically jump to the answer, I think the hardest thing to understand about life is not what it's made out of, which is what we've classically focused on, but how it processes information. And how did that arise? That's the hardest thing in the origin of life, and I also think that's the key to recognizing life on another world, is not recognizing some substance, but recognizing the fact that information processing has been going on. So that's what I'm trying to put some thoughts on together, and this is where we are, and this is the date, and this is me, and this is where I'm from. That's all pretty straightforward. So the first part of this is, how did life get started? In some sense, it reminds me in a kind of whimsical way of a mathematical problem of NP problems, which you're easy to show that you have the answer, but it's very hard to get the answer from first principles. Very hard to solve, but very easy to verify. If you have a solution, you can check trivially that it's the solution, but getting the solution is actually very, very exponentially hard. We have a solution. We know that there is a solution to the problem of life. In fact, if we didn't have it here, I would conclude it was impossible, right? But we have it here, so there must be a solution. Proof by one example, yeah. Yeah, exactly, as far as we can tell, yeah. And we care. But it's very hard to solve. So this is a good starting point.
Was that the undiscovered Earth then?
That's Mars, billions of years ago. That's why it's smaller.
God is smaller too, right?
God's smaller, and the origin of life is smaller.
It was less significant.
Yeah, it happened faster. So what do we know about the origin of life? Well, if you go to the literature, there's lots of papers. Each box or circle represents papers or a class of papers. We don't have a consensus theory for the origin of life. It's not like, it's even worse than theoretical physics. You know, you get string theory and all these other theories here. We don't even have a standard model that people want to attack. You just have a bunch of competing ideas, and the only way to approach the question of the origin of life on other worlds, which is what I'm supposed to be doing, is by analogy. So I put down all the theories and say, would they work somewhere else? Well, all of them suggested for Earth would work for Mars, right? So that's one reason why Mars is such a good target. Some of them would work for my other favorite worlds, Europa and Enceladus, but not all of them. So we don't really know how life got started on Earth. We're searching for it somewhere else. Many of the theories for how it got started on Earth would work on the other worlds, but not all. So now I'm going to try to start pushing in the information end of things and make the distinction between the physical chemistry of life and the information content of life. So the two approaches to the origin of life in some broad category are, first, life is a structure. It's metabolism first, it's reactions first, and then it learned to encode information and reproduce later. So it's chemistry first, hardware first, software later, okay, to use the language of computers. The other approach turns that around, is that first, life learned to encode information and reproduce that information, and then with that knowledge, it built up knowledge on how to build things and how to eat. So this is eat first, think later. This is think first, eat later. Okay.
Why are you doing both at the same time?
Well, you could say life started simultaneously, thinking and eating, but now you've doubled the difficulty of the problem. Yeah, right. So you could say that somehow you simultaneously start in both, and who knows? But this is what the thought is, and there's war between these two camps, the metabolism first versus the replicator first. The RNA world thinks they've solved the problem in this area. They think first there was the RNA molecule, which was able to reproduce. It can do autocatalysis. It can also store information. And so it was the first life, and the notion of an RNA world is very popular now in the origin of life. But then the metabolism people say that's a bunch of garbage, because if you do any sort of prebiotic synthesis, you can't make RNA world. All you can do is make chemistry. You make reactions. So you can make things that are metabolizing and processing and structures. Chemistry is what comes automatically, not information processing. So we're at an impasse. But this then, I want to just put a little plug in here for why it's useful to talk to the IT department. Because the language and the mindset of computer science has really revolutionized the way we think about the origin of life and how life works. And it's given us a vocabulary to express things much more succinctly and clearly. Like stating that life is like a computer, it's made up of software and hardware. It's a sentence that's very easy to say now, and we all understand what it means, that there's a component in our body that's hardware, like proteins, catalyzing reactions, or the Calvin cycle, making energy out of sugar, right? That's all hardware. But then there's also information coded in the genome, which is really software. And this distinction was really not, even though the basics was understood, it was not crystallized in the field of the origin of life until computer science developed this logic and approach to thinking of things. Yeah.
That's a TRS-80.
Yeah. Well, I was looking for something a little earlier, but this is what I could find, right? Yeah.
Paper tape.
So instead of teasing you, I have a more serious question. Turing compiled his software into circuits, into hardware, and ran it there.
Yeah, okay.
So the two don't necessarily have to be split this way.
It's not clear that it was a computer.
That's even right, yeah, because all the computing was in his head.
Right.
He made a circuit like the old plugboard analog computers.
Right.
So there is no computer, it was compiled away.
You could imagine computers that don't make a distinction between hardware and software. Like
there's not even a computer
the things
there.
you see at Babbage's computing engines, there's no software or hardware, it's all on the wheels, right? So you can imagine that. That's not the way computers work, and since this paradigm has now been taken up by biology, we've embraced
Not
it.
quite with 3D printers now, we can make a mechanical computer that does it basically the Turing way.
Yeah, and this is an interesting.
Sorry, I don't want to sidetrack.
I want to open it up because when we're searching for life elsewhere, we're assuming the same software-hardware dichotomy that we have in modern computers and we see in life on Earth, right? We think there will be a separation between software and hardware. And this has important practical implications for how we search. And for example, this is a floppy disk. One reason I put floppy disks here is because they illustrate an essential feature of information storage is that you don't want the shape of the storage media to depend on the information written on it, right? You think, well, that's kind of trivial and obvious. You wouldn't want the shape of a floppy disk to change if you wrote a different file on it, right? If you think of a protein, there's a molecule that has information in it, but the shape of the molecule depends on the information in it. So you change the information, you change the shape of the molecule. The wonderful thing about DNA is that it is like a floppy disk. You can change the information content in the DNA, and the physical structure of the DNA doesn't change. That's actually highly unusual for a chemical molecule, where the structure doesn't depend on the information in the molecules that make it up.
They're all proteins.
No, DNA is not proteins. DNA is not a protein. DNA.
You said the DNA, the information stored.
The DNA, right. But I'm making a general statement that life on another world, we are embedded with this paradigm that life is going to have some kind of molecule that, like DNA, can store information without changing its shape. So we have the software paradigm distinction, and it's in our thinking. Maybe it's wrong.
The idea of information is clearly central to this. But for instance, information can exist on a light beam. For instance, you can have a resonant cavity, and the modes of that resonant cavity contain information.
Okay.
And it's possible to conjecture a form of life in plasma, which is nothing more than the modes of energy in a resonance system. Okay, so whereas information is central, how the information is encoded may cloud our view of what we could see.
Okay, I accept that. You're way beyond what we're able to do in terms of the search for life, and I'll come to that. If you think of this in the abstract as you're doing, sure, there's lots of ways you can encode information, lots of ways you can have physical systems that act on that information, right? The point I'm making here is there's 2 points. One is the connection intellectually between the origin of life and biology now and soft computer science. That's sort of a history of science point of view, perspective. The other point is this distinction between hardware and software, which is real in current computers and real in current biology as we know it, and is the basis of how we're approaching the search problem. So we'll come back to that. But the point that this distinction is a convenience because the companies that make software are different than the companies that make hardware. It's just a historical accident. It's not a fundamental property. I accept that. And I also accept the fact that you could mix them in many different ways too. But this is the way that the non-computer people think of computers, and this is the way we think of life now, right? So let's go to the hardware for a minute. The important observation that we're using about the hardware is that it's what I call Lego hardware. What I mean by Lego hardware is that biology is not just a random collection of stuff from the CRC. It's certain blocks, certain building blocks stacked in order, in long chains, and the complexity comes not from the diversity of the blocks, but from the combinatorial possibilities of the stacking. This is a fundamental property of the big, giant polymers of biology. The power and the diversity comes not from having a large number of building blocks, but from stacking them in a large number of ways. And that's the way Legos work, right? You got a box of Legos, and there's only a few different types in it, but you can build anything you want out of it in principle by stacking them in different ways. And the large structural information containing polymers of biology are the proteins, which are made out of 20 left-handed amino acids, DNA and RNA made out of nucleotide bases, a handful of them, and polysaccharides made out of sugars. Those are the Lego blocks of life on Earth. And I've argued that this is likely a common property of biology, as well as mass-produced children's toys, throughout the universe, for reasons of efficiency and economy and selectability. This is likely to be a property. Darwinian evolution will push life toward this parsimonious utilization of certain molecules. It'll be more efficient because each one of these molecules that you use, you've got to control and produce and stock up and so on. Your body's got to process all these amino acids. If you had 100 amino acids instead of 20, you've got a bigger problem. Right? So you don't want to have to deal with too many constituents. It's a supply chain problem. You don't want to have to amplify your supply chain problem any more than is required to get the job done. And that's why this will be a common property of biology everywhere. Minimize the supply chain problem. And we're going to use this principle in the search for life. I'm going to get to this in steps follow. So here is the pictures of these molecules. These are the 20 amino acids. Here are the purines and pyrimidines and the sugars, and add in a few fatty acids, and you've got it all. Now, one of the most amazing results so far is that we can basically make all this stuff in the lab for free. If you just take, famous Miller-Urey experiment. I didn't have a picture of the Miller-Urey experiment, so I pulled one from our lab where we're doing the same thing in a Titan-like experiment. We take nitrogen and methane, put it in a flask, put a spark, and all sorts of complex organics are made. If you add water and ammonia, you find a lot of these things are made. A lot of things that aren't these things are made too, but these things are made. So you make the building blocks of life and a bunch of other crap that is not part of the building blocks of life. And it's the bunch of other crap that's an important part here. What you need to separate the building blocks of life from all the other crap is information, right? And so the hardware of life comes for free. It's sort of like being able to create computers by just putting a spark through a rock, silicon rock, and have computer chips fall out, right? It's that wonderful that we can do that. The chemistry is that clever that it can do it that way.
Organic building blocks throughout the universe?
Not yet. I will, but not yet. Now, I'm just doing a retrospect of these that life uses. So a lot of these, of the 20 amino acids, about 10 of them, you can get for free by just running this experiment. Well, the famous case was done by Stanley Miller in 1953. Miller-Urey, it was his PhD thesis, where he did the spark discharge with water, nitrogen, ammonia, methane, water, spark, and found that many of the amino acids in life were produced. Subsequent to that, about 10 years later, the discovery of these same amino acids in the Murchison and Orgay meteorites falling in from space. So they're in space, you make them in the lab. It's clear that we've now discovered them in the interstellar medium, some of them, glycine, for example. So you get them for free. You get some of these basic building blocks for free. Life just makes them. Like I said, it's like finding silicon chips out in the middle of the desert. They're not programmed, they're blank, but they're there.
What percentage of it is useful life components and what percentage is
It's
other
very
things?
small. The percentage of it that maps onto the biomolecules of life is small. I don't know what it is off the top of my head, but it's small, maybe a percent or two. So it's mostly black gunk. It's mostly tar. It's mostly useless from a biological point of view molecules. But the stuff that's needed is there. And when Miller, that was 70 years ago now, 60 years ago. When Miller published this result, it was quite a stir. And the origin of life field thought that it was just a matter of a few years before they'd be able to have life in the lab. They had the same positive outlook that fusion researchers had back then. Just a few years. And they've had the same success that the fusion people have had in producing that process in the lab. But it was a big thing because we didn't, in retrospect, you realize this is just the stuff. This is the hardware. What's missing is the software. What's missing is the information required to go from these bricks of life into a self-replicating system that stores the information that assembles the bricks, right? And I think we now realize that that's the hard problem. Making these chemicals is not the hard problem. That's the easy problem. The fundamental problem is a software problem, not a hardware problem. The universe gives you the hardware problem. It's a gift from the periodic table, the hardware. But where do you get the software? We're clueless as to yet. So now my interest in this is not to create life in the lab, although that'd be good fun, is to use this understanding to base a search for life elsewhere. So my day job is searching for life. Okay, that's what I report into work. What are you here for? Well, I'm supposed to be searching for life. Okay, you're in cube 3. And so the approach we're taking, I like this quote. I shall not today attempt to further define it, but I know it when I see it from the Supreme Court, that source of much wisdom, is basically the approach we take to life too. When we go elsewhere, we've got an example here, we'll know it when we see it, and that's the approach I'm taking, and you'll see that over again. And the practical implications of that approach is, what does life need? Don't try to define it. Just try to say, what are the environments on Earth which we see that it survives in, and then go look for those environments. A very empirical, theory-light, observation-rich approach. And liquid water is the critical environment, so hence the so-called follow the water. Now, the next slide should have been earlier. This is the software of life. This is an example of what I mean when I say life has software. This is the common words in the language of all life, comparison of the conserved sequences in the 16S RNA. This is data from James Lake at UCLA. And you can see that in these different organisms, from humans down to Giardia and corn and E. coli, all have the same sequences in the conserved regions of their RNA. This is almost like there's a tag, I am from Earth, I am from Earth, I am from Earth. Every life form on Earth has this sequence. Pay no attention to the viruses for now. That problem is to be solved in a different seminar. But the point is that there's all life on Earth runs the same operating system. You can recognize it just by looking at it. Or a metaphor I use when I'm talking to students is, all the books here are written in English. All of them.
All we have is the fungi.
Oh yeah, well fungi, we're way above fungi. Let me see, fungi is in here, but this is E. coli down here. That's a bacteria, right? And so here's, pardon? Up here? Thanks. Oh yeah, fungal, it says over there. There's a code here for, in English, animals, fungi, plants, protozoa, archaea, all the same. And I'll show soon a tree of life. But this is the software. The other thing we know about life, this is sort of a random collection here of things we know about life, it's a Darwinian system. And this is, again, traces back to the information content. Life is a material system that shows reproduction, mutation, selection, based on information coded in the software, manifested in the hardware, and then mutations on the software. Okay, now, you could also say that you could have a Lamarckian system, where the hardware is the information, and it mutates, and it evolves. Right? There's no Lamarckian evolution, as you may remember, is erroneously attributed to Lamarck by the Anglophiles who want to privilege Darwin's discovery, but nonetheless, we'll take that as labels. Darwinian evolution is there's a separation between the information and the instantiation. So the hardware is different from the software. The software programs this hardware, then the hardware interacts with the environment, and based on that, there's selection on different software. So that's the Darwinian paradigm, which turns out to be true. There's no way Darwin could have known that was true. He didn't know anything about DNA, but it turns out it is true. There is a separate DNA and a separate genotype and phenotype. In a Lamarckian scheme, there is no separation between the genotype and the phenotype. In a sense, the RNA world is a Lamarckian world. If the molecule changes, its offspring change because its information content has changed. Whereas in the Darwinian world, if something changes, the DNA doesn't change, and so the offspring, if you lose your leg, your child is not born without a leg. In the Lamarckian world, if you lose your leg, your child is born without a leg. And the RNA world is a Lamarckian world, right? And it's not against the laws of physics to imagine life forms that are Lamarckian, where the information is what you were suggesting earlier, or somebody was suggesting earlier, where the hardware is the software, and they're not physically separated. We're not going to go there, because I would never get a proposal funded that was based on that, right? And this will come.
So such a life form would transcode its physical realization as a way of reproducing. It would literally traverse its physical structure as the recording of its state.
Well, I don't know about that, but let me express it in the way I do know, which is the RNA world. I should have put up a slide of the RNA world. The RNA world, the RNA molecule, which is like a DNA molecule, has information coded in it, but it also acts directly like a protein in making catalyzing change. So it is both a genome and a phenotype. So it interacts in the environment directly, unlike DNA, which is shielded from the environment. Our DNA does not interact with the environment. Our proteins interact with the environment. Right? The DNA instructs us how to build proteins, and the proteins interact with the environment. And then based on that interaction, some DNAs win and some DNAs lose, and those mutations are selected for. That's the Darwin paradigm. And the RNA world is not a Darwinian world. It's a Lamarckian world because the DNA itself, the RNA, itself interacts with the environment. And if there's a change in that RNA due to some interaction with the environment, if it loses a nucleotide or gains one, then when it reproduces, its daughter offspring will have that same change. Right?
You're essentially saying that DNA is like hardware and the RNA like hardware plus software?
No, I haven't translated into those words.
Well, maybe you should, because at least for this community that might help.
Yeah, it's more, I think it more in terms of Darwinian versus Lamarckian.
No, I understand.
And we don't need to dwell on that too long because we're going to ignore the Lamarckian logic, but I put it out there for completeness.
Can I at least put the footnote on that? You excluded viruses, but if you know viruses can read and write to our DNA, like read and writing to our CD-ROM, I joke.
Right.
At that point, the two are much more similar than you think.
Let's stay focused.
Viruses are, I think, are not a very interesting case because they will not, we don't think we'll find them by themselves. Okay, let's go back on to now I want to turn to searching for another example of life, what I call a second genesis. As you can see, there's a lot of confusing issues with life, understanding it, we don't know its origin. What's the way forward? Well, one way forward has been sort of try to make it in the laboratory. And this is where computers come in and computer simulations are playing a role here. And people are making the RNA world. There's a group in Boston and a group in San Diego that are making the RNA world and trying to create self-replicating molecules from the ground up. And of course, people are doing biology simulations in the computer as well. Same point, trying to understand some of these questions that we've been talking about. As you can see, since I chose a hammer for my icon for the laboratory, I'm not a lab guy. So I don't work in the lab. Another approach, of course, is to wait for them to call. This is SETI. Just wait for them to call. But I'm not very good at waiting for phone calls. So the approach that I'm involved in and I'm going to talk about now is go try to find it on another world. So we want more data, go find it. Go find another example. And that we would learn a lot about these questions by having more than one example of life. So a lot of the questions might become reinforced, or our answers that we think are specific to Earth life might become reinforced if we find the same answers in other life, or they might be disproven if we find a different example. So why do we have any idea to think that there could be a different type of life? And this is a question that biochemists like to ask me. They basically say, well, look, we're right here at the top of evolutionary selective optimization. Any way you go, it's downhill. There can't be another solution. Now, that's not the words they use, but they basically are saying there's only one maximum, one optimum in the entire phase space of biochemistry, and we're it. We're on that optimum, and they're certainly correct. Any direction you look from that optimum is downhill steeply, right? Now that doesn't prove that there's only one optimum. It proves that we're isolated, that the optimum we're on is isolated, but there may be an optimum way over here for all we know, right? And biochemistry, the textbook is 500 pages. There's got to be more than one optimum in that many pages of textbook, right? That's my argument. Biochemistry, life is maybe a localized phenomenon, and there may be several cases where it sticks out. Now that doesn't mean that we're looking for something like silicon life or something strange. This is organic biochemistry. And again, I like to express it in terms of these simple concepts. How would that compare to Earth life? I'm still postulating that at the basic level, the life would be composed of carbon and water. Not getting beyond that, not the plasma life or Fred Hoyle's black clouds or Robert Ford's neutron star life yet. Love to see that, but one step is just what can we find in carbon and water? So my argument is that at the bottom, it's going to be the same. Life is going to be carbon and water on Earth-like worlds because carbon and water are the available resources. At the top, life is going to be the same. Big fish will eat little fish. Sunlight will be the energy source. CO2 will be the carbon source. Ecologically, it will be like us, and elementally, it will be like us. Where there's a possibility for difference is in the inside, is in the biochemistry, in the information processes which turn this into systems that interact like that. Question?
Water is your solvent. I mean, in the conditions that we're accustomed to, it's going to be water, but with higher pressure, it could be CO2 or ammonia.
Right. The practical reason for restricting ourselves to water is because when we look in our solar system, which are the worlds that I can get to, there's only two liquids accessible in our solar system. One is water, and that's on Earth, Mars, Europa, and Enceladus, and the other is liquid methane, 95 Kelvin on Titan. So those are the two liquids I care about. You're right. Theoretically, you can imagine other liquids that are more suitable than liquid methane, that's for sure. But remember, my job is to go out, do missions, and search for evidence of life. So it matters what's available to search for. There's no place where we have liquid CO2. There's no place where we have ammonia, which would both be good candidates for alternative liquids. The only other liquid we have is liquid methane, which is a pretty poor choice, but that's the one I'm focusing on other than water, as I'll say in a second. So I'll kind of develop this theme further, in that this information, interior information, is what we can expect to be different in water worlds that are still like the Earth. And we know how to search for this. This is the tree of life. We talked about it already. An alien is an organism not on this tree. This is a little bit of why we would search for this. Why are we searching for a second question here?
In the Urine-Miller experiments in the junk,
Yeah.
were there any other amino acids?
Well, yes.
Or where nucleic acids form?
Well,
Okay.
amino acids for sure. There was a lot, and I'll come to that. There's a lot of amino acids formed that are not part of the set used by life on Earth.
So I was going to ask about exotic species on hot sea vents, and I see you have extreme thermophiles.
Yeah, they're not exotic. They're only exotic in the sense that people that live in Thailand are exotic. To them, it's perfectly normal. They're on our tree of life. They use DNA, RNA. If you were to grind them up in a blender, they wouldn't look any different than if you ground up your next door neighbors in a blender in terms of the fundamental biochemistry. Don't do this experiment at home. But the point is, this is all the same stuff. We have not found anything not on our tree of life. Okay.
Where those branches meet, which is the
common
That's the root
what you
right
were
there.
talking-
That's the root of, if you trace back these trees, and how you draw the trees depends on various assumptions and things. It's not a unique mapping. But if you trace it back, there's a common ancestor somewhere around here, the last universal common ancestor. This is one of the amazing results of biology over the last 50 years, is the tree of life and the common ancestor and the relatedness of all life on Earth. And again, it's hard to place viruses on this tree, but clearly they're related. Liquid, I'm going to speed up a little bit. Where to look is the next step. Earth, Mars, Enceladus, water, as we were talking about earlier. Titan is the other world with a liquid, but the liquid's not water. So the question, unfortunately, in our solar system, none of the environments we can access are likely to be habitable, which means we're not going to likely find anything that's alive. What we're going to find is dead microorganisms, death. So we're not doing a search for life, we're doing a search for death. The distinction is between something that's dead and something that was never alive. Right? If you're driving down the road, there's a rock on the ground. Okay, it's a rock, and next to it there's a dead rabbit. You know there's a big difference. The rabbit was once alive, right? And that's what we're searching for. We're not going to find anything that's alive. If something's alive, you can grow it and you can reproduce, and that's how Viking tried to search for life. We don't have that option. We have to search for basically dead bodies. But dead bodies are very, very interesting. And so another way to phrase that question is, if we find organic material, can we tell if it was ever dead, if it was once in a while? So the question is what's different about organic matter that was alive versus organic matter that was not alive? And the poster child for this difference is the amino acids in proteins. In proteins, in life on Earth, the proteins contain L, left-handed amino acids. In the meteorites and in the Middle Urey experiments, we find both left and right. And this gets to your question. What was in the gunk that wasn't biologically useful? All the right-handed amino acids. Now, a purist would say, well, life does use some of these D amino acids in other things. Some of them are poisons, and some of them are used in cell wall linkages. But in proteins, it's only the left-hand side, left ones. Right? So that's an important observation. Life selects, chemistry doesn't. And this is part of the lingo, so the Lego principle. So expressed graphically, you can think of a distribution. Say you have a clump of organic matter that came from a meteorite or from a Middle Urey experiment. You'd expect it to be a smooth distribution in some sort of generalized phase space. Molecules with similar properties would have similar concentrations because they're being made by chemistry. Chemistry doesn't care. So similar properties means similar chemistry. Similar heats of formation, blah, blah, similar degeneracies, and so on. Biology cares, and so molecules with very similar properties can have vastly different concentrations. The concentration of L-alanine is very different than the concentration of D-alanine because one is used and amplified by biology and one is not. Right?
Is there the potential of conjugate life, which is to say symmetry? It would be possible to build a life form which used the other handedness?
Yep. You're at about 5 slides ahead of me. But we'll get to that. Hold that thought.
Okay.
So this is a distinction between biology on Earth and chemistry. And you can imagine alien life having the same principle. It uses certain molecules and not others, but it uses different molecules. So that's the basic approach that we want to take in searching for life. Now, let's refine that a little bit more. And the way to do it, I think, is to focus on amino acids. I'll come back to that as to why they're our focus here. But this is a diagram from a paper we just finished talking about this problem and pointing out the distinction between the simple amino acids and the complex amino acids. So this is a plot of the number of distinct molecules that are possible as a function of the increasing complexity of the amino acids. So here, the bottom, where there's one carbon atom, there's only one amino acid, glycine, and that's it. If you've got you're telling me I've got a one carbon atom amino acid, guess its name. I get it right every time, glycine. But if you were to say I've got a 10 carbon atom amino acid, guess its name, there's like 9 factorial different versions of it, right? So I'd be guessing for a long time. Right? So the number of possible combinations goes up very steeply as the number of carbon atoms goes up. That's carbon atom, C number. Right? These simple ones, the C1, C2, C3, there's only 2 or 3. These are also the ones that are made by prebiotic chemistry. So everybody's going to use them. Everybody in the universe that has amino acid chemistry is going to use glycine. You get it for free. It's the only one of its kind. If you've got a C1 amino acid, that's it. And there it is. And probably same up to C2, C3. When you get to about C4 or 5, the choices become astronomical. You just can't, you have to pick them. So these are the ones that are used by Earth life. Earth life uses up to about C4, then a couple C5, 6, and then a C9 and a C11. Right? Now, when you get up to those large numbers, the chances that life, so the way we think of this is life inherited these from the prebiotic soup, these simple ones, and then it invented these using evolutionary processes. So it made a selection for these. Right? The odds that other life would select the same from the combinatorial diverse set is vanishingly small. and many of them have the same properties. If what life needed up here was a sulfur-rich amino acid to do stabilization of proteins as a function of temperature, well, there's lots of sulfur-rich amino acids. So the chance that it would pick the exact same one is very, very small. So our approach is look for amino acids and look for, did I go backwards? No, let me go forward. I'm going to skip this and go right now to the amino acids. So let's focus on amino acids. So here's Earth's, let's look at all possible amino acids as some big set. One interesting thing is life only uses 20 of them. And those 20 split into a 10 that we got for free and a 20, and then the second 10 that we invented, life invented. So you can imagine a strange biology that uses the same 10 but has a different in the second set. And some very important work by Philip and Friedland showed that alternate sets of 20 can span the phase space of charge, size, and hydrophobicity as good as Earth sets. Right? So they are expressed mathematically, they are as good a basis vector set for constructing things in this space as the 20 we have. Right? So there's, well, I'll get to that in a second. In this diagram, they're all left-handed. We're just thinking about differences in the type of amino. Now, you could also imagine, but this is harder to imagine, that doesn't use even the simple 10 and makes them all. Stranger biology has no overlap with Earth's set. This is what we'd expect. Overlap in the simple ones, divergence in the complicated ones. The simple ones, you get them for free and they're unique. The complicated ones, you don't get them for free, you've got to make them, and there's a combinatorial vastness in the number of choices each time you make one. Right? And there is the chirality, there's the handedness. So not only does not only does life have to choose the amino acids that it's going to use, it has to choose whether it's going to drive on the left side of the road or the right side of the road. Right? Once you've decided, then everybody has to fall in line. You go outside, all the cars here are going to be on the right side of the road, we hope. Right? Somewhere else, they could all be on the left side of the road. Don't go to Italy. They'll be on both sides of the road. But pardon? Not necessarily. Some L-amino acids are toxic, but a lot of medicines now are made out of, D-amino acids are toxic. Some of the birth defects associated with phthalimine, I think, were due to D-amino acids at the time. Right. Right. Ibuprofen also has some chiral mix in it as part of its function. And now, this is the industrial sector that's the most invested in D-amino acids is medicine, because they will make medicines out of D-amino acids. Because if you're taking a pill that's made out of L-amino acids, the proteases in your saliva and your stomach will tear it apart. Whereas if you take a pill made out of D-amino acids, those proteases are ineffective. So you can take orally drugs that would otherwise have to be injected, and they last much longer against the degradation processes in your body because they're the wrong kind of amino acids. And if you make them with the same functional shape as their L counterparts, they have the same efficacy medically. So it's a big industry of producing D-amino acids for medical applications. And we'll come back to that in terms of making mirror life. They're the ones that are on the road. We just have to ramp up their technology a couple orders of magnitude, and we can make an entire E. coli that's the mirror image of itself. Now, if you notice, there's a rabbit over here and not over here. And this is the point. In the original Alice, well, in the original Alice, there wasn't complete symmetry in the two worlds. There was, as the physicists would say, there's a parity violation. The rabbit was in one world and not in the other. Now, we think that there's complete symmetry here, that if you took an E. coli and made its exact mirror opposite, or image, all the L amino acids become D, all the D sugars become L, the DNA curls the other way, everything is the mirror image. We think it would operate perfectly. But we have no data to support that. The only data we have on this is taking this up to the level of catalysts. So groups have made D catalysts out of D amino acids and shown that they are specific for D peptide bonds. So that step has been shown. But taking it to the level of a whole organism, we haven't done.
To replace other 10 amino acids to create stranger biology.
People have tried to incorporate and there are a few natural occurrences of small instances. There's 2 other amino acids that are sometimes used in proteins and sort of in the fringes. So if a purist would say this is really 22, not 20. And people have come up with other coding schemes besides the coding schemes that incorporate these, that can incorporate strange amino acids. That's right. There's a lot of manipulation in the, we can now manipulate the transcription process and vary it. What I'm suggesting is building the mirror image of the whole process.
Have there been considerations of the potential biological hazards of creating the genesis of conjugate life?
Well, yeah. This is ICE-9, the biological equivalent of ICE-9, right? You know that Kurt Vonnegut story? ICE-9, this would be the biological equivalent, right? Could be. But, you know, it's, I go to this quote from Feynman on his blackboard. What I cannot create, I do not understand. We're in that situation with life. And we know that life works. We have a solution. The simplest thing that we can make is the mirror, that's different, is the mirror image of that solution. Now people, you see in the press, they created life. Well, what they did is they took the genome of an organism and then without really knowing what it does, just duplicate it, sort of like taking a Xerox copy of it, and then stuffed it into an organism in which they'd remove the genome and then boot it up. Yeah, that's impressive, and I'm glad it's a paper in Science, but it's not creating life by any stretch of the definition of the word create. Right? This would be, creating a mirror life would be creating life. You would have created a self-replicating, functioning organism that has no ancestor on Earth. You've created it de novo. And
Is there enough one-handedness in the feed chain that it could still eat?
Yeah, well, microorganisms can eat both handedness of amino acids. They have enzymes, racemases, that turn it into the one they like. So you can train microorganisms to chow down on D amino acids. They turn them in, they racemase them to L and use them in their proteins. That's a well-known tool. Higher organisms like us, we detoxify them. We don't use them, we throw them away. But microorganisms who are poorer and can't afford to, they turn them into L.
The right kind of sugar you can eat, but it disassembles it, basically, right?
Yeah, the sugars I'm not so sure of.
No, it's less the right kind of sugar is okay, it's not toxic.
Yeah.
But the thalidomide one is toxic.
Yeah. So the last thing I want to talk about is where are we going to put these approaches to test. My favorite word right now is enceladus, the plume coming out from a pressurized water pocket full of organics. We want to fly through it, take samples, and search for amino acids. So if you say, what is the biomarker we would search for? What is the molecule, as I said, the Lego block that we would search for? Amino acids are my choice because we know that they occur naturally in comets and asteroids. We know that life uses them. And we know that life on Earth has made 2 choices in their use, choosing the chirality and choosing the distribution. And we know that at least chirality, there's no reason why life elsewhere couldn't have made that choice differently. There's a right-left symmetry. And we know that in the distribution, we're pretty sure that 10 of the 20 at least are chosen arbitrarily. So we want to explore that. Now, I want to end with a thought, which is the dog chasing the car. Right? There's a dog chasing the car. And you ask the dog, well, what are you going to do if you catch the car? Right? This is an important question. And a little kid, I was giving a talk like this, not this talk, but a talk to the public at Death Valley a couple months ago. And a 12-year-old girl said, what are you going to do if you find life on Mars? And I know some people snickered in the audience, but I said, that is a profound question. It's the dog chasing the car problem. And generally at NASA, we think nothing about it. We don't think at all about what are we going to do if we find life on Mars, and it is a second genesis of life. That it represents the first example of a life form that is not on our tree of life. What are we going to do? Well, I think this brings up the question, which I'm not going to dwell on, but the moral status of alien microorganisms. If we find a second genesis in our solar system, I contend that then there are 3 sets that warrant moral consideration. Humans, of course, are a set that warrant moral consideration. Life itself is a set that warrants moral consideration. Not equal to everyone in the set, but we all appreciate that there's something special about life, right? In the case of humans, we not only appreciate there's something special, but we want to assign sort of an equality within that set. We don't do that in life, but that doesn't detract from our sense of its specialness. Now we find another example of life. That's a new set, life 2. So I would argue that microbes, which score low moral status based on any of the criteria that are assigned for moral status, feeling pain, complex behavior, communication, cuddly little smiles, whatever it is that you like about life that makes you have moral regard for it, are low on bacteria. And in fact, you probably kill thousands of them when you wash your hands and brush your teeth, and I encourage you to continue doing that. But what if we found them on Mars? They would have high moral status as being the sole representatives of the set Life 2.0. That's it. It's the last buffalo taken to the microscopic level, and the diversity taken to the highest possible level, a completely different type of life. So what does this mean? Well, you can say this is just silly philosophical ramblings. But it's not. I actually argue that it has immediate implications on how we explore these worlds. And the conclusion, which I won't dwell on, but I developed in a couple papers, is the robotic and human exploration of Mars should be done in a way that is biologically reversible. We must be able to undo, again, the computer metaphors entering into our language. My favorite one is the undo, Control-Z. I think it's a wonderful concept to, imagine, I would like to have that in my life, Control-Z, let's Control-Z the whole day, everybody, right? Aren't there times when that would just be the thing to do? So far, we have contaminated Mars, but our contamination is reversible. We can Control-Z. An example of irreversible contamination is the rabbits in Australia. You can't undo that. But if the 2 rabbits that had started the Australia colony had been dropped off in Antarctica instead of Australia, you could undo that contamination. You just go back and find them, 2 dead rabbits, not far where you left them, right? So everything we've done on Mars so far is we can reverse it. And if we discover a second genesis of life there, we should hit the undo button until we've cleaned it up.
How would we undo our contamination of Mars?
By going to all the landing sites after Viking, when we stopped sterilizing spacecraft, and collecting all the hardware and bringing it back to Earth, or sterilizing it rigorously in place. We stopped sterilizing spacecraft after Viking. And so Curiosity, which just landed on Mars the year before last, carried with it 500,000 viable microorganisms from Earth. That was its bioburden, allowable bioburden. Sounds like a lot, but the dishes in my apartment have more bacteria per square centimeter than that on them. So it's not clean and it's clean, it's not sterile. Most of those microorganisms would be killed upon exposure to the Martian environment, UV. But the ones inside the spacecraft, shielded by metal or plastic, will only be killed over timescales of thousands of years by cosmic rays. So we can estimate now that there's about 2 or 3 million Earthlings on Mars, dormant inside various spacecraft. We know where they are, we know where all the pieces are. They're in the litter that we put on the surface. And as long as they're inside, and as long as Mars is dry and cold, they're not growing, they're not doing anything. If we wait long enough, they will die, but that's thousands and thousands of years. So if we find that Mars has life and that it is a second genesis, there's a conflict there between our contamination and that life. But so far we can reverse that.
Suppose we send a human to Mars, then what?
Still reversible. That's why that picture's there. As long as their activities remain on the surface, they are living in a biocidal bath of UV radiation. So the only things that aren't killed instantly are things that are inside plastic or metal. And so as long as we keep track of all the bits of plastic and metal so that we can pull them up if we have to, we can do it. So in principle, a human base is reversible as well. Now, if you were to say drill deeply down into an aquifer and contaminate that, that's not reversible. Right? The surface of Mars is doing us a favor, being incredibly inhospitable and being bathed by UV light down to 190 nanometers, which is very biocidal. biologists would love to have that kind of sterilizing capability in their clean room. Right? Question here?
This flyby mission to one of the moons, are you expecting to carry laboratory equipment that will allow you to do full molecular characterization, mass specs, and genetic sequencing and all
We'd
that?
like that, but what's there now is able to do this. What's on Cassini, it's flown through the plume, is a mass spec that goes up to 100 AMU and has a resolution of 1 mass unit. It's sort of like a $3,000 RGA, residual gas analyzer, that might be in a plasma physics lab. Right? It's great, but it's not, a state-of-the-art GCMS. I doubt if we'd be able to get that. That's why I'm focusing on what single molecule do I want to focus on, amino acids. So focus the analytical capability on that. It'd be great to be able to characterize all large molecules, all macromolecules, as you could do with a sample return to Earth, which is what we really want to do. But in situ, I would just focus on the amino acids. We just measure them. And then maybe we'd only pick a few of them, a few of the 10 and a few of the 20. You don't go through the whole set of all possible amino acids. That's too hard. But you might look at glycine. It's almost certainly going to be there. And then you might look at, say, alanine and serine and see, are they both of the same chirality? On Earth life, the alanine and the serine, the two simple chiral ones, would both be L, dominated by L. That would be pretty cool if they were both dominated by L on Enceladus. It'd be even cooler if they were both dominated by D. Right? Then you might look for some complex sulfur-containing amino acids to see if life had invented something out there at high carbon number. And that, if we could do that, I would die a happy man. That's enough.
How much bigger would the spacecraft have to be to do the complex
Analysis, you would really...
It's not so much a function of bigger, it's cost more. Right now we're aiming for a discovery target, which means we're restricted to small instruments 1 and 2. And we're competing with people who want to do other things that aren't related to the search for life, like go look at rocks or sulfur volcanoes on Io and so on. So we have to have a solid science story, and it has to be cost effective. So we can't get there on the first step. Now, if we fly through and find amino acids, and there's a higher concentration of D, then we can write our own check for the next mission, right? So it's a step at a time. Question here?
Like this, isn't it more possible for finding a second Genesis on Enceladus or on Europa rather than Mars because of the exchange of material between Mars and Earth?
Yes, that was something I skipped, but that's a very good point, and that's this arrow here, which is increased chance of life not related to Earth life. So there may be life on Mars, and as you're suggesting, it may turn out to be us, carried there, maps on our tree of life. And the further we get from Earth, more likely what we find, if we find anything, is that it's alien. Of course, since Titan wins on that, hands down, because we're not related to anything that lives in liquid methane, not by any chance at all, right? But these other worlds are water worlds, so it's possible that life from Earth contaminated these worlds, or vice versa, or they all shared contamination from somewhere else. So it may be that we'll find life here, here, and here, and it will map right on our tree of life. It will not be a second Genesis.
Will we be looking for optically active stuff?
Yeah, that's what I would want to do on this mission, is look for algae. We don't call it optically active anymore. That's a historical term from how it was first discovered. Chirality, left and right, was discovered by Louis Pasteur because of the rotation of light. And so it's been called optically. But that, in the biochemical world, that terminology is sort of considered quaint and old-fashioned. Now we just call it we call it chirality. So we will definitely want to detect chirality and look at the difference, because all amino acids that life uses except one have these two chiralities. Glycine is too simple, so it doesn't have chirality. But alanine and serine, for example, very simple amino acids still, but they have chirality. So those would be ones we would definitely look for and see if one chirality dominates over another. In the meteorites, they're about the same.
One reason to ask the question the old-fashioned way, with lasers, you might be able to do remote detection. How far can you go? Do you know?
And microwaves. There was a paper in Nature about a year ago showing microwave detection of chirality from the interaction, from the scattering of the microwaves. And to me, that was a epiphany.
But how far out?
Well,
in terms of signal reasonable.
They weren't very far, but
Okay,
that's
so you
just
have
a
to go
technical
more.
problem. To me, that was, I had a suddenly blinding flash of light. It was my road to Damascus moment. That is how the tricorder works. Right? Because all these years I've wondered, how did the tricorder work? So here's Spock in orbit, and he's detecting life on a planet below him. You say, well, what is the property of life that they're detecting so they can differentiate it from non-biological matter? What is the property of life that could be detected remotely? And the property is, life is chiral. It selects only chiral. I forget what you need, but whatever you need, it's just a matter of the engineers to work it out now. Yeah, I've got the paper on my computer. I could pull it up.
Because those are hard to do generally.
Yeah. But those are just technical difficulties.
Okay.
The conceptual solution is now in hand.
Fine.
We have
That's what I mean.
a solution to how a tricorder works.
Do I understand that when you say chirality and its optical significance is the absorption spectra of circularly polarized light is different for the two spiral directions? Is that correct?
That's correct. And that's how they were discovered by Louis Pasteur.
So we have there's a whole additional diversity to the absorption spectra based upon circularly polarized light.
Yes.
Can't be seen with single direction light. You need any circular.
Yeah. But this is not true just of those molecules. Calcite crystals will do the same thing. They preferentially transmit polarized.
It's called Andreev scattering.
Yeah, it's
Quarter wave plate.
Or a
Quarter wave plate does that.
Okay.
That's not enough.
Yeah, but anyway, yeah, that's the same thing. You can and pardon?
Quarter wave plate is often.
Okay. But
there's a complication there.
birefringent crystals, that's what they do. That's the correct answer. That's the non-biological equivalent. Birefringent crystals.
But then you.
Okay.
Birefringent is basically having two frequencies, but it's K and omega.
If you assume that Enceladus doesn't currently have life, but did, and if it's been dead for 1 billion years, at ordinary temperatures, which we assume it has to be because you have liquid water, natural racemization occurs.
Right.
At what point do you lose the ability to discriminate that it was once chiral?
Yeah, that's a good question. 1 billion years is a long time at 0 degrees. The good thing about Mars is it's been frozen, its deep sediments have been frozen at -60, which is much better than 0 for preservation. So I would not be optimistic about finding stuff on Enceladus because 0 is not a very good storage temperature. Mars in the permafrost, the deep permafrost, I would be because -60 will get you to the age of the solar system. So if it's dead a long time, but the good thing about Enceladus is the samples are fresh. They're coming out of the water right now. So I'm hoping that it's only been dead a few hours.
Chiral is pretty stable physically.
But it does racemize over time. It's been measured. In fact, it's used as an age dating on teeth and things like that. So.
Why can't you have a coexistence of both handednesses of life in the same ecosphere?
Why can't we all just get along? We don't. It's an observed fact.
No.
Well, I think we can hypothesize systems where an equilibrium could exist.
This would be an interesting question for computer simulations. If you could do an accurate simulation, are there solutions where you can have two systems that compete at this fundamental level? So what are they competing for? They can't eat each other, but they compete for access to sunlight, access to water, and access to physical space.
Actually, Chris, if you had come to dinner, you could ask the woman who could answer that question is coming to dinner.
Oh, but unfortunately, Gene, much as I'd like to go to dinner, the telecon, Japan is so many hours different. This is a U.S.-Japan mission. We have a telecon at 6 to start designing the sample return capsule, so I'm going to have to miss dinner. But your point is that people are doing these computer simulations. And that's one of the things I was saying. We could do things in the computer that we can't do in the real world yet. Eventually, we'll be able to make mirror life, and we'll put E. coli left and E. coli right in the same box, hopefully tightly sealed, and see if they can coexist.
Poison each other.
My conclusion is,
Two life sources, two genesises. That's much harder than one.
well, I would actually think that what happened is we've had
How do you get the
multiple.
second from the first?
Well, if you have a cross-genesis, you have a process by which you transform one into the other.
If I was writing a story,
We can do that, but
if I was writing a story, I'd have multiple genesis. We think it's easy. It started many times. But life is a winner-take-all game.
Yeah.
That at one point, somebody eats everybody else out of the game. And I think that will show up in the computer simulations. That if you do the simulations correct to nature, you will not have two systems. One will eventually out-eat the other and drive it.
Up to 90%. Right now, it's 90% typically.
Sorry?
Left-handedness in biology is more like 90%.
It's not 100%.
No, it's 100%.
No.
In the sense, I don't mean that 100%
depends on
of
the
our amino
detail.
acids are L, but I mean we are all part of the same
Yes,
system.
true. But if you analyze the brain, about hand-for-handedness by 90% is left and 80%.
Okay, that's a different thing. I'm talking about, the question was, could you imagine more than one tree of life? Let's generalize it not just to R&D amino acids, but go back to this tree. Why is it not the case that on Earth, why is it the case on Earth all life is related? Why couldn't you imagine that we see on Earth two separate biospheres, biological systems that trace back to separate, distinct origins, in the same sense that we have English and Chinese. They are both written in paper, they're both in books, and they both compile human knowledge, but there's a fundamental difference in the logic of the way they encode information. In this world, all the books are in English. There are no books in Chinese. And so the question, I'm generalizing your question, why couldn't you imagine that on Earth you had a second genesis of life growing over here and our genesis here, happily sharing the Earth, plenty of ecosystem space for all. We don't see that. My argument would be that at one time there probably was, and it's just a matter of competition. And eventually one type dominates and excludes the other out of all niches and drives it to extinction. It's a winner-take-all game. And I bet if you do the simulation right, you find that in the computer simulations.
You introduce Google Translate and the happy coexistence of Chinese. What I'm saying is if you introduce two species which cross-translate the ecosphere, you could get systems that
That requires an outside agent.
Well,
An altruistic outside agent. Yeah, but again, these are all questions that could be addressed in simulations. We can't address them in nature because we can't even reproduce this, much less make an alien one to compare it to. The mirror life would be the first example of us trying to do that. Create a mirror life and compare it. So I'm all for this line of inquiry. I think it's a fascinating question. To me, it would be highly surprising if life started off this way and just developed this way. I think almost certainly at some point there was more diversity at the whole life level, and then it got winnowed down. I mean, there's kind of a weak parallel to this in Homo sapiens, and at one point there were multiple species of humans. Now there's only one. You could take the view that it's a winner-take-all game there, too. You know, I think much as we might think share and share alike is a good moral philosophy, it may not apply to nature in the raw.
The detection of chirality that you showed there sounds like it requires access to a sample.
Yes.
But
Or
so
this
you need to
remote.
have, so it's something that you have to directly detect. But how would you translate that into a remote sensing problem?
Well, this is what you were saying earlier. It may be there are methods
Like
now
index for solar
discussing
planets, for example.
that can tell the difference between these type remotely. One technique that's just developing is Raman spectroscopy, and the other is this radio frequency wave, basically looking at.
Radio station, Alaska.
Well, if you can fly up to it, you can get pretty close to Enceladus now. I don't know, I haven't gone
Same
back and looked at the
technology
Star
can be
Trek.
used.
I don't know how far the tricorder's range was. It certainly worked from orbit. So
Well,
that,
that one is for against Soviet missiles, so it
Yeah.
can go pretty far.
So you can
left-handed
cite that as a technology feasibility
Yes,
precedent?
exactly.
The tricorder exists?
I'm going to put in a patent application.
Well, now you explain my kids, I mean, they have exquisite hardware, but they have very buggy software.
Well, evolution is all about improving the software.
Right.
Yeah, going back to your kind of moral equation a little bit, you kind of distinguish between human and microbe, but kind of our current understanding of humans, we're probably a pretty significant percentage made up of microbes.
Yes.
So
Yes.
kind of a couple of things come out of that. If we discover microbes someplace else, L2 microbes, might we assume or have to assume that there's a higher level order of life until we can disprove that in terms of our
Yeah.
model equation? And the second question is there any way by looking at microbes to detect whether there's higher order life associated with them?
Let's go back to the tree of life. That's a good question. The first I've thought about, the second I haven't. So if you go back to the tree of life, let's see, it's in here. Here it is. These are microorganisms, the red and the blue trees. They are both inside of us. The archaea and bacteria live inside us. Mostly bacteria, but the archaea do as well. One of the curious features is that only this branch, the red branch, causes infection. So only the red states are dangerous, the blue states are not. Why, we don't really know. Now, for most of Earth's history, there was no multicellular organisms. It was only these guys. So they could certainly imagine an ecosphere, an ecosystem with only microorganisms. Your other question is more interesting. Is there any, if you had samples from here and here, could you know that this existed? I don't think so, because if you could do it the other way around. If you had these guys, you would know that these guys existed, because these guys have have metabolic diversity. Up here, the diversity, to oversimplify, is only morphological. These are all the same. They're all aerobic. They're all eukaryotes stacked. They just look different. But here, they don't look different. They all look the same, roughly speaking. But they have different chemistries. Some are like oxygen, some don't. Some use sulfur, some use iron. So there's all the metabolic diversity in the biosphere is in these two branches. So looking at these guys, you could see that they must have had ancestors or cousins or related that had more diversity than they do. But looking at these two, I don't think you can infer that one. I'll think more about it, but I haven't thought about it before, but just thinking about it now on the spot, I don't think so. Pardon? Oh, right. Well, sometimes, I go to the middle of nowhere and give a talk, and people, you're from NASA. Don't you guys have aliens hidden in your secret storage locker? And I say, yes, indeed, and I'm happy to show you a picture of them. There they are. These organisms are definitely not on our tree of life, and that is our definition of alien. So,
Slightly more serious, back to the viruses, they are left and right-handed, or at least some of them are. So these may put the real spin on your story.
Yeah, but not in the amino acids. Remember, in the DNA, the chirality becomes a little mushy. And it's not even clear the chirality is important to DNA, because you unzip it before you do anything with it.
It gets complicated, yes, I agree.
But the amino acids, that's another reason why I like amino acids, because there, the chirality is clear, you can't mix it, it's, even the viruses need...
But the viruses can do something.
They can
Because they can rewrite your DNA and then the others get screwed up too.
But when they're controlled, they make proteins. They're still made out of left-handed amino acids. So that's much more universal in biology than the chirality in DNA. And the chirality in sugars is interesting, but not as important as in proteins. So that's why we focus on amino acids and proteins as the key molecule. That's one of the arguments why the one biomarker I want to search for when we get to these other worlds is amino acids and this signature, and I think that's this is it. amino acids are for a lot of reasons, are the best molecule to search for first. Now, once you find them, DNA is a lot more interesting than amino acids. But if you're doing the first search, if we find amino acids and we find a signature, like I said, we'll then get the money to do the real mission where we send a big lab up there.
Just concerned you tossed out the virus story. So I'll
Yeah.
talk to you afterwards. I don't want to sidetrack you on it.
All right. Okay, well, I think it is afterwards, so thanks for your attention.